Okay, so hi everybody. My name is Mariana, and I'm very glad to be here to present my infographic for the Points of Pride Research Day. Okay, so I'm going to talk about in vitro intestinal culture. The first question is, what is in vitro intestinal culture? Well, it's a technique that I am using in my research and is able to mimic the intestinal mucosa. In this technique, we are able to culture organ or pieces of tissue for a period of time in the lab. Okay, the second question you may ask, what are the steps that we need to do in order to perform this methodology? So first, we need to have a donor animal. In my case, I am interested in swine enteric disease. So the donor animal that I have been using in my research are healthy young pigs around 5 to 6 weeks of age. Important to highlight here that the animal should be healthy and not showing any clinical signs of diarrhea. After you collect a portion of intestine around 10 centimeters from the animal, we bring to the lab, and then in the lab we open the intestine. First we wash in order to remove the feces, then you open the intestine, and after open, we cut in little pieces of 2 by 2 centimeters that you can call explant. So each explant will be then individually placed in a 6-cell well plate with median, and after that we can transfer the cell plate with each explant in the incubator for a period of time. So at the end, what you can say about the explants is that each explant is a living good outside an animal. Okay, we are maybe wondering what are the advantages of this method over the other in vitro methods such as cell culture and organoids. Well, the in vitro intestinal culture keep the tissue architecture and they also keep the cell diversity. In addition to that, is able to reproduce the inflammatory response and they also keep the gut microbiome. Additionally to that, the in vitro intestinal culture is in line with the principles of 3Rs. So first, reduction. By using explants, so one donor animal can give to us several explants. And with that, the number of animals needed for the study is less than would be in an in vivo study. The second thing is refinement. So in some cases, you need to challenge the animal to study the disease. In this case, we can challenge the explant and not the animal. So at the end, we're gonna minimize the suffering and the stress of the animal. Besides that, when you compare in vitro intestinal culture with in vivo experiments, in vivo trials cost much more because it requires high costs with animal care and facilities. Okay, last thing, applications. So we can use explants in order to study host pathogen interactions, mostly in the early time point of the infection, which is hard in vivo models. The second application, we can use explants in order to test of non-antimicrobial compounds, such as prebiotics, organic acids, in order to see if they are effective or not, and this can be an alternative to antimicrobials in swine production, but also in animal production. So with that, I would like to thank you for your attention, and I hope you could learn something about this interesting methodology. Thank you.